Hello class. For this lecture, we will be talking about glycolysis. And so glycolysis process is set up in two different stages. The first stage is glucose is trapped and destabilized and cleaved into two interconvertible 3-carbon molecules generated by cleavage of 6-carbon fructose. So the first, or also known as the investment stage of glycolysis, uses the energy from two molecules of ATP. So in that thought process, it's just like trying to create a business. Sometimes you require money in order to make money. And so for this process of glycolysis, it requires ATP in order to generate more ATP. And the products of this stage are two molecules of GAP, which stands for glycerolaldehyde 3-phosphate, which is drawn right here and listed here. For every molecule of glucose entering the pathway. So once again, you have one molecule of glucose and it ends up generating two molecules of this glycerolaldehyde. But to make this glucose into this glycerolaldehyde, we have to sacrifice exactly two ATP in order to generate this molecule. And then on the second stage, when you have that two molecule of GAP, now you have 2 times as much of ATP that is being generated. So now just think of it, you sacrifice 2 ATP and you got out of it 4 ATP. So you're getting more ATP than was originally sacrificed in order to generate more and more energy source. And so this is the basic 2 stages of glycolysis. The first one is the investment stage, and the second one is the payoff stage, where now we're generating ATP, and at the end, you have pyruvate at the end of the glycolysis phase. So we're going to investigate each of these steps. So the first step is you have glucose and you have a phosphorylation of glucose. So that hydroxyl group that is part of glucose will now have a phosphate group attached using this hexokinase enzyme, and this enzyme requires magnesium in order to be active, in order to take ATP and take one of the phosphorus group from ATP and attach it to the glucose molecule, where you have the results of ADP. And so the magnesium actually shields the negative charges of the phosphoryl group in ATP to make the terminal phosphorus atom an easier target for nucleophilic attack by the hydroxyl group, mainly the oxygen of the glucose. And so basically within this reaction, you basically have glucose going in and you have ATP going in. And then from there, with magnesium ion, you have a generation of your product, which is just ADP, and glucose with the 6th position of having that phosphate group. And so the binding of glucose to the enzyme actually elicit a structural change within the enzyme to now be able to hydrolyze the ATP from being ATP to ADP. But without that glucose molecule, the ATP binds to that hexokinase enzyme, and it does not actually hydrolyze the ATP. So that glucose is very important for that enzyme to actually start to hydrolyze that ATP, and that phosphorus group will be attached to that glucose, and then you have that ADP being exported out. And so this is just showing you a little bit more detail of what's happening within that magnesium activity. It actually shields these negative atoms, and then now that oxygen, or you can think of the hydroxyl group within glucose, can now do a nucleophilic attack to that phosphorus atom, and now releasing it, where now you just have two phosphorus group, which is ADP, and the phosphorus group that it loses goes now on to the glucose. And so now for the second step is the conversion of glucose 6-phosphate to fructose 6-phosphate. This is a reversible change, so we can still go back to glucose once we get into the fructose, but it utilizes the enzyme phosphohexose isomerase, and so it also uses that magnesium atom that helps shield things. And what also things you should notice that as far as the comparison of the two carbohydrates, this one you have a ketose, and then what it was originally was an aldose type of carbohydrates. And so how is this happening? First, we have this ring opening that happens, which is actually catalyzed by histidine residues that's within the enzyme. And now you have that ring that is open, and then we have a glucose type of residue within the enzyme that act as a base to deprotonate the hydrogen on the second carbon, and thus that molecule get protonated by outside protons that's in the solution. And then from there, that intermediate is going to want that hydrogen back from that glucose residue within the enzyme, and thus releasing another proton. And now we're in the fructose type of form for the carbohydrates, and you see that we have the ketone right here, because we have a carbonyl and two carbons on both sides, and then we have the closing of the ring, where now we're in the fructose ring, and now this is also catalyzed by the histidine residue within the enzyme as well. And now going to the third stage, where we have phosphorylation of fructose 6-phosphate. And so once again, we have this hydroxyl group. It's the same type of mechanism, but we have a different enzyme. Now we have phosphofructokinase, which is short for PFK1. It uses the magnesium cation to help shield things, and it utilizes ATP as a source for a phosphate group that will be transferred on to that fructose 6-phosphate to produce the fructose 1,6-bisphosphate. So it got the 1 because of that 1 position of the phosphate group, and then 6 because of the 6th position of the phosphate group. The phosphate group that goes to this 1 position came from another source of ATP, where there's hydrolysis of the ATP to generate ADP. And so this is an irreversible step. And so now we can no longer have the form of glucose anymore. And so this particular step is known as the rate limiting step of glycolysis. And so this enzyme, its activities is increased when the cell's ATP supply is depleted. So that means we don't got that much ATP around, so we need to invest more ATP in the investment stage in order to make sure we get that more of that payoff to start replenishing more and more ATP within our bodies. But also, if we have too much ATP around, then the enzyme is actually inhibited. And so it will see that it doesn't need to work as hard because we got plenty of ATP, so we don't need to invest as much in stage 1 of glycolysis to produce more ATP since we have an ample amount around. And then we go to the 4th stage, which is the cleavage of fructose 1,6-bisphosphate. And you have that cleavage that's happened between carbon 3 and 4, and it produces 2 molecules, glyceraldehyde 3-phosphate, and also the dihydroxyacetone phosphate. And notice that for both of these carbohydrates, one is an aldose and this one is a ketose. It utilizes the enzyme aldolase, and once we have these 2 molecules, eventually the dihydroxyacetone phosphate will end up being the glyceraldehyde 3-phosphate, where we end up having 2 of these instead of just 1. And so how is this happening? So once again, we have this ring opening that is happening, and then we have lysine that acts and does a nucleophilic attack to the carbonyl of the carbohydrate once the ring is now open. And then from there, we have other amino acids that acts as a base or a acid, and thus we have some rearrangement that occurs, and thus some dehydration that happens where we release water, and thus having this form of what is called a protonated Schiff base. And then from there, once we have this form, we'll start having more of the amino acids that can act as a base to deprotonate and take up one of the hydrogen, and then thus making a cleavage happen, and thus releasing our first compound, which is the glyceraldehyde 3-phosphate. And then from there, we have isomerization that occurs, and thus now we can have the second product be released after hydrolysis, and you have the dihydroxyacetone phosphate group, and then you have your original enzyme ready to have some more of the fructose 1,6-bisphosphate to start breaking it down once more into the 2 products of the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate. Then we go into stage 5, which I was hinting at before, where when we have that dihydroxyacetone phosphate, it needs to be converted into that glyceraldehyde 3-phosphate. And the enzyme used for this interconversion of the triose phosphate is called, in short, TIM, but it's called triose phosphate isomerase. And that concludes stage 1, which is the preparation type of stage of taking glucose, convert it all the way down to the GAP, which is also known as glyceraldehyde 3-phosphate, and it just sacrifices 2 ATP in order to generate that. And once again, you have your glucose, it gets phosphorylated, and then from there you have the glucose goes into the fructose stage, and then it gets phosphorylated again, and that's that rate limiting step. And so once you add this fructose and have the 2 phosphate groups attach, you can no longer get into the form of glucose ring after this stage. And then from there, you have cleavage and eventually get into that glycerolaldehyde 3-phosphate. Stage 6 is the oxidation of glycerolaldehyde 3-phosphate, where it utilizes inorganic phosphate, and it uses a nucleotide, NAD+, that actually gets reduced and oxidizes this glycerolaldehyde. And then eventually we have the phosphoryl group that is attached to the original glycerolaldehyde 3-phosphate. And it utilizes the enzyme glycerolaldehyde 3-phosphate dehydrogenase. So how is this happening? You have glycerolaldehyde 3-phosphate that enters the enzyme, and the cysteine residue within the enzyme does a nucleophilic attack onto the carbon, now having that glycerolaldehyde 3-phosphate group attached to the enzyme, and then now the NAD+ does oxidation onto the molecule, and now it is reduced to NADH, and we have the carbon chain gets oxidized. Now we have this carbonyl, and from there NADH is released and it gets replenished with brand new NAD+. Also, what goes into reaction is the inorganic phosphate. And from there, that inorganic phosphate does a nucleophilic attack to that carbonyl, and now attaches the phosphate group to the glycerolaldehyde 3-phosphate and releases it from being attached to that cysteine amino acid residue. And now you have, as far as your final product, the 1,3-bis phosphoglycerate. And so this is just a breakdown in text what is all happening. So in general, you have GAP, which is once again that glycerolaldehyde 3-phosphate. It binds to the enzyme by a nucleophilic attack by the cysteine amino acid, and that actually creates this thiohemiacetal. And then from there, that NAD+ goes and oxidizes the thiohemiacetal to produce the NADH. And then from there, the NADH goes off and the NAD+ comes in along with the inorganic phosphate, which does a nucleophilic attack on to the thioester and thus releasing the 1,3-bis phosphoglycerate. And now the enzyme is brand new again and can start this process all over again. And then the 7th stage is the phosphoryl transfer from 1,3-bis phosphoglycerate to ADP. And so now we're starting to pay off. Because remember, we have 2 of these molecules because we started off with now 2 of these glycerolaldehydes. So there is 2 1,3-bis phosphoglycerate. And so thus, when we generate this ATP, just note that this is 2 ADP that is coming off. Because we have 2 of the 1,3-bis phosphoglycerate. And so it utilizes the enzyme phosphoglycerate kinase along with that magnesium cation. And what do you think this magnesium cation is for? It's for shielding, just like it has been doing for the previous steps. And so, as I mentioned, that magnesium cation is shielding the negative charges on ADP, and now it can freely do a nucleophilic attack to this phosphorus atom and pick up the phosphate group and now have successfully have ATP. And now that 1,3-bis phosphoglycerate is no longer having one of the phosphate group, and now you have just 3-phosphoglycerate. And now from there, step 8, we have the conversion of 3-phosphoglycerate to 2-phosphoglycerate utilizing the enzyme phosphoglycerate mutase, along with having magnesium cation. And thus, you'll see that that phosphate group moves from the 3rd position to the 2nd position of this carbon chain. And so what happens within this enzyme, it has histidine that is activated by having a phosphate group that is attached to it, and thus it gives away that phosphate group, and another histidine amino acid residue actually does a nucleophilic attack and takes up one of the proton from the 2nd carbon, and it gets replaced by that phosphate group. Then from there, that amino acid that lost its phosphate group comes a little jealous and like, okay, I want it back. And so when it tries to take it back, it actually doesn't take back the exact same phosphate group that it originally had. It ends up taking it up from the 3rd position phosphate group. It doesn't matter. It's like it just wants its phosphate group back. Whether or not it's the original one or some form of a phosphate group, it wants it back. And so thus the one that the histidine that is protonated, it said, okay, I don't need this proton. And so it gives it back to the glycerate type of carbon molecule. And then from there, now we have that phosphate group that is now on the 2nd carbon, and the 3rd carbon no longer has the phosphate group attached. And everybody is happy again because this histidine is back to its original state, and that activated histidine that had the phosphate group that's attached now is happy and can start this process all over again. And then you go into the 9th stage, which is the dehydration of 2-phosphoglycerate to phosphoenol pyruvate. And so once again, we have this dehydration is happening where you have a release of water, and it utilizes the enzyme enolase. And then from there, once we're in this form, the last step in glycolysis is to transfer of the phosphoryl group from the phosphoenol pyruvate to ADP, which is catalyzed by pyruvate kinase, which requires potassium and either magnesium or manganese cation. And so just note that most likely when you have this magnesium cation, it's going to act as a shield. And then from there, it will help promote the actual exchange and transfer of the phosphoryl group. And so how is this happening? Once again, you have that magnesium that is acting as a shield. And now the oxygen can now do a nucleophilic attack to the phosphate atom that is part of the phosphoenol pyruvate. And now we have ATP that is released. And then that magnesium and the potassium still act as a shield, and then the molecule goes through tautomerization, you finally have your pyruvate. And so this initial form was the enol form of pyruvate, and with the tautomerization that happens, it goes into this keto form, which predominates at pH 7. And so thus this concludes the payoff stage of glycolysis. Remember, we had that glycerol 3-phosphate group, 2 of them, and then it goes to transfer of phosphate group to be attached on to the first position of the carbon, and then from there it starts having some payoff happening and transfer phosphoryl groups to ADP to generate ATP. We go through another stage of preparation where it's some interconversion of having that phosphate group from the 3rd position and now move to the 2nd position. Then you have dehydration that happens, and then it's finally at a stage where now it's on the last stage of donating its last phosphate group, now generate ATP, and you finally have 2 molecules of pyruvate, and overall in total there is 4 ATP that is generated. And so as far as the fate of the pyruvate, remember we have glucose, it goes to glycolysis and it produces NADH and also 2 molecules of pyruvate. The pyruvate goes to fermentations and produce lactate, ethanol, and carbon dioxide. Or it can go through an aerobic condition where it gets oxidized and thus produce an NADH, carbon dioxide, and then it goes through a citric acid cycle to produce carbon dioxide and water. And so this process, with at least with the citric acid, we will discuss in the next few lectures that is coming up. And that concludes the process of glycolysis.